Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I hope that you will be fine and enjoying good health. I am Dr. Muhammad Ali Hashmi, and we are studying the subject Introduction to Biochemistry with a course code CHEM2114. This is lecture number 21, and in this lecture we are going to study about significance of lipids in biological membranes. The main concepts we are going to learn in this lecture would be definitely significance of lipids in biological membranes, with special emphasis on membrane proteins. There will be a brief introduction about membrane proteins. There are some proteins which are linked to the lipids in the membrane, so we have to look at them for lipid functions in the biological membranes. The next main concept would be lipids which are present in the membranes. What are their functions and how they are present in the membrane, what is their arrangement, etc. And then we'll look into the fluid mosaic model for the membranes. These will be the main concepts of this lecture. Now we come to the composition of some biological membranes. Biological membranes are composed of proteins associated with a lipid bilayer matrix. So what is the main composition of biological membranes? They are composed of proteins, and those proteins are associated with a lipid bilayer matrix. Bilayer means that the lipids in the membranes, they have two layers, and one of them is pointing outside to the cell, and the other one is towards the inside of the cell. And the proteins are associated, they are attached to these lipid bilayers in specific ways, which we'll learn in the upcoming slides. So the lipid function of these proteins consist of complex, the lipid fractions of these proteins, they consist of complex mixtures that vary according to the membrane source. So we'll soon look at a table where we'll see that different membranes, according to their source, they have different composition. So in different types of membranes, the percentage of lipid fraction and their complexity and their structure that differs in different kinds of membranes. There is not one type of cell in a body, or in a human body, for example. There are so many different kinds of cells, and each cell has their own kind of membranes. And then you know that eukaryotic cells have organelles, membrane-bound organelles. So those membrane-bound organelles also have membranes, and their structures are, of course, different than the cell membrane. So all these different membranes, they have different kinds of lipids and proteins. That really depend on the function of those membranes and specific position of those membranes. Membrane proteins carry out the dynamic processes associated with membranes, and therefore specific proteins occur only in particular membranes. So as I said that because these proteins which are present in the membrane, they have specific functions. They sometimes let the substances pass through the cell membrane, sometimes they stop those substances, sometimes they act as gates. So in different membranes, proteins will have different functions. So those different membranes will have different kinds of proteins according to their function. Next is protein to lipid ratios in membranes. These ratios vary considerably with membrane function. So as we just studied, that different membranes have different ratios and different kinds of proteins and lipids. Similarly, protein to lipid ratios in membranes also differs in different membranes. Although most membranes are at least 1/2 protein. So this is a generalized statement that most of the membranes have at least 1/2 of their structure that is protein. But the actual percentage really depends on different kinds of membranes. For example, the myelin membrane, which covers the myelin cells, it functions passively as an insulator around certain nerve fibers. So around certain nerve fibers, there is a membrane called myelin membrane, which functions as an insulator. There is prominent exception to this generalization, which we just said that 1/2 of the membrane is protein. So in myelin membrane, only 18% proteins are present. So this is an exception. This is not included in that generalization which we just made. So let's have a look at the table which is said here, and it's presented on this slide. Here, this table presents compositions of some different biological membranes. For example, if you look at the plasma membranes in mouse liver cells, human erythrocyte, and amoeba, so they have different percentages of proteins, lipids, carbohydrate, and protein to lipid ratio is given in this last column. And if you look at human erythrocyte, it has 49% protein, 43% lipids, and 8% carbohydrates as well. Similarly, if we look at other kinds of membranes, for example, mitochondrial outer membrane that has 52% proteins, 48% lipids, and some 2 to 4% carbohydrates as well. And as we studied, that myelin membrane has only 18% proteins and it has 79% lipids. You know that what is the main reason why it has so many lipids than proteins? We just studied in the previous slide that what it does, it actually acts as an insulator. So lipids are very good as insulators. There are many marine animals which have thick layers of lipids which protect them from the cold environment. So that is why these cells or these membranes have so a higher percentage of lipids in them. Now we are going to discuss about the composition of membranes under the following main sections. That is, number one, we'll discuss about membrane proteins, and then we'll have a look at lipid linked proteins. What are the lipid linked proteins and what are their functions in the biological membranes? So the first one is membrane proteins. Membrane proteins are operationally classified according to how tightly they are associated with membranes. So they are basically classified on the attachment or on their attachment to the membranes and on their tightness of the attachment. So the first one is integral or intrinsic proteins. Integral or intrinsic proteins are tightly bound to membranes by hydrophobic forces. So these proteins, they are bound to the membranes by hydrophobic forces. What are hydrophobic forces? The forces due to water repelling nature. We'll have a look at the figure on the next slide. And they can only be separated from them by treatment with agents that disrupt membranes. So they are bound, the intrinsic proteins are bound to the membrane so tightly that they cannot be simply isolated or separated from the membranes, but they can only be separated by treating with the agents that disrupt the membrane, mean that just destroy the membrane, and then the protein is separated or dissociated from that membrane. So they are very tightly bound to the membranes, and these include the agents which can disrupt the membranes. They include organic solvents some detergents, and cheotrophic agents, that means ions that disrupt water structure. So these kinds of chemicals can destroy the membrane and separate the integral or intrinsic proteins from the membranes. So here is the structure which was just said. This is a protein which is an intrinsic protein or integral membrane protein, and what this structure is representing, we're just going to discuss here. The protein is represented by its surface diagram, which is colored according to charge. So red color means negative charge, blue color means positive charge, and white means uncharged. So these red areas are means that these portions of the protein have negative charge on them. Blue areas mean they have positive charge, and white color means that this portion is uncharged. Tightly bound molecules of dimyristoyl phosphatidylcholine are drawn in space-filling form with carbon green, oxygen red, and phosphorus orange. You can see that the membrane has some tightly bound molecules with it, which are shown in the space-filling model, and the different colors are shown for the different atoms. For example, these green colors are carbon atoms, these red color atoms are oxygen atoms, and then these orange atoms are phosphorus atom. So these dimyristoyl phosphatidylcholine molecules, they are tightly bound on the protein. You can see that these molecules are attached to this protein, and you can note that how the lipid tails closely conform to the non-polar surface of the protein, thereby solvating it. So these are the tails of the lipids which are attached to the protein, and they are the reason, because of them the protein can be solvated by a solvent due to these non-polar parts of the lipid attached to it. Integral proteins tend to aggregate and precipitate in aqueous solvents, aqueous solutions, unless they are solubilized by detergents or water miscible organic solvents such as butanol or glycerols. So in aqueous solutions, what happens that integral proteins, they aggregate and they precipitate. They do not get soluble in aqueous solutions. Aggregate means they just get together to form a kind of a cluster and precipitate out, and they are solubilized by detergents. So detergents, you know that detergents have both polar and non-polar parts in them, so they can easily solubilize them, or they can be solubilized by water miscible organic solvents such as butanol or glycerol. So you know that organic solvents can dissolve the lipids portions, so that is why these integral proteins can be dissolved by these organic solvents. Some integral proteins bind lipids so tenaciously that they can be freed from them only under denaturing conditions. So as we already studied on the previous slide, that these integral proteins are so tightly bound to the membranes that they can only be separated by the solvents that denature, that disrupt the membranes. So some integral proteins are so tightly bound that they can only be freed from those membranes under conditions in which proteins are also denatured. So you cannot really isolate them in intact form. If you try to isolate them, they will be denatured or destroyed. So you cannot get those proteins separated from the membranes. Then we have peripheral or extrinsic proteins. First were the integral or intrinsic proteins, and the second is peripheral or extrinsic proteins. They are dissociated from membranes by relatively mild procedures, so they are not as tightly bound to the membranes as the integral proteins. That is why peripheral proteins can be dissociated from membranes by relatively mild procedures, and that leave the membrane intact, such as exposure to high ionic strength salt solutions. For example, if you expose these proteins to high ionic strength solution, for example 1 molar NaCl, which has a lot of ions in it, sodium and chloride ions, and metal chelating agents, or by pH changes you can even isolate or separate these proteins from the membranes. Now, all biological membranes contain integral proteins whose locations on a membrane may be determined through surface labeling. It is a technique. Surface labeling is a technique by which we can locate, or we can determine the position of a protein, integral protein in the membrane. So in this technique we employ agents that react with proteins but cannot penetrate membranes. So those kinds of reagents are employed in this technique, the surface labeling, which can only react with proteins but they cannot enter the membranes, so that they cannot disrupt the membranes. So that's how we can determine the position of integral proteins on the surface of the membranes. For example, if an integral protein on the outer surface of an intact cell membrane binds antibodies elicited against it, so there is an integral protein which binds antibodies, and it is towards the outside of surface of the cell. It's not inside the cell. But a protein on the membrane's inner surface can do so only if the membrane has been ruptured. So if the antibodies are attached to the membrane's inner surface protein, that means that the membrane has been ruptured. Otherwise, those antibodies cannot enter the cell. They have to bind to the protein which is pointing towards the outside of the cell. So membrane impermeable protein specific reagents that are fluorescent or radioactively labeled may be similarly employed. So that is why we use the agents which cannot enter the membrane like antibodies, and they only attach to the outside proteins of the cell, and those reagents are fluorescent either, or they are radioactively labeled so that we can detect them where they have attached, on what position on the membrane they have attached, and that position will be the location of the intrinsic or integral protein, because those agents will definitely attach to the proteins only. Some integral proteins are exposed only to a specific surface of a membrane, whereas others, known as transmembrane proteins, span the membrane. So that means that some proteins, some integral proteins, they are only present at certain locations, at specific locations on the membrane. But there are other proteins, other integral proteins, which are known as transmembrane protein. They span the membrane. They are present throughout the membrane. So this is the difference between transmembrane protein and some specific integral proteins. However, no protein is known to be completely buried in a membrane. What does that mean? That all have some exposure to the aqueous environment. So proteins are not completely buried in the membranes, but their heads or some of their structure is protruding outside to the membrane, so that they have contact with the external environment. So outside the cell is the aqueous environment. So proteins, they are pointing outside. That is why they can interact with different substances which are present in the extracellular environment. Now, biological membranes are asymmetric in that a particular membrane protein is invariably located on only one particular face of a membrane, or in the case of transmembrane protein, oriented in only one direction with respect to the membrane. So what does that mean? That means that membranes are asymmetric. They are not they cannot be divided in equal halves. Why? Because there are some proteins which are only present at certain locations, For example, you divide this slide in half and one protein is present on the right side here but not on the left side, so these two halves will not be symmetric halves. So in that sense, some proteins are present at specific locations, and some other proteins, they are present on one face, either on the outer face of the membrane or the inner face of the membrane towards the cellular environment. So that is why the membrane is an asymmetric structure. It is not a symmetric one. Yes, so now we have a look at the diagram of a plasma membrane. Now if you look at this structure, as we first studied, I believe on the first slide or at the very start of this lecture, that membranes are composed of lipid bilayers. So you can see that this is a lipid bilayer. This is the first layer of a lipid, and here is the second layer of lipid. So you can see that these are the polar heads, the blue colored, these blue spheres, they are polar heads of these lipids, and these tails are the non-polar long fatty acid chains. Now these bilayers, their non-polar heads are towards each other, and their polar heads, the non-polar tails are towards each other, and polar heads, they actually face the they face the outside environment. For example, if you consider that this is the intracellular environment and this outside is extracellular environment, so you can see that in both cases the polar parts or polar heads of the lipids, they are facing the outside world, and the non-polar parts are attracted towards each other on the other side. So this makes a lipid bilayer. We'll have a look in the fluid mosaic model later on about the function, how these two bilayers, these two layers are arranged together, and what are their specific functions. And now we have a look at the integral protein. Here is an integral protein. There are other proteins, lipid link proteins, peripheral proteins. So you can see that they are very tightly bound to the lipids in there. And we also studied one more thing, that these proteins are not completely embedded in the membrane. You can see that they are protruding outside the membrane. They're not buried or embedded inside the membrane. Some of their portion is protruding out of the membrane. So and then those portions which are protruding out of the membrane, they can perform different functions. They can act as gates for allowing certain substances to enter or exit the cell, and similarly they can perform other different functions. And you can also see that there are some oligosaccharides present and glycolipids, and yeah, the carbohydrate components of glycoproteins, yellow beaded chains. These yellow beaded chains are the carbohydrate component of the glycoproteins. And glycolipids, what are glycolipids? They are here, they are shown in the green beaded chains. They occur only on the external face of the membrane. So this is external face of the membrane. This below portion is the intracellular environment or internal face of the membrane. So these glycolipids and glycoproteins components, they only occur on the outside of the cell side of the plasma membrane, so outer face of the plasma membrane. Next we come to the lipid linked proteins. First we studied about the proteins themselves, now lipid linked proteins. Lipids and proteins associate covalently to form lipid linked proteins. So what is the nature of the interactions between these lipids and proteins and lipid linked proteins? They form covalent interactions. So they are bound chemically through covalent bonds. That is why these are strong bonds, and These lipid portions anchor their attached proteins to membranes and mediate protein-protein interactions. So the lipid portions of these lipid linked proteins, they anchor their attached proteins to the membranes. Their proteins are embedded or attached inside the membranes, and they mediate protein-protein interactions. You can see that this is a lipid linked protein, and they are responsible for different protein-protein interactions, and they are attached to the membrane, as you can see here. Proteins form covalent attachments with three classes of lipids. Now, what are those three classes? The first one is isoprenide groups, such as farnesyl and geranyl residues, and then fatty acyl groups such as myristoyl and palmitoyl residues, and the third one is glycoinositol phospholipids. Let's have a look at these three different kinds of classes of lipids. The first one is prenylated proteins. A variety of proteins have covalently attached isoprenoid groups, mainly the C15 farnesyl and C20 geranyl residues. What is isoprene? Isoprene is a 5-carbon hydrocarbon, and it is the main chemical unit from which many lipids, including cholesterol and other steroids, are constructed. Isoprene is the basic unit of many different biologically occurring molecules like terpenoids and lipids and some cholesterol and other steroids, etc. So this is the basic building block, like a cell is the basic building block of a body. Isoprene is the basic building block of these bigger molecules. So have a look that isoprene is a 5-carbon unit, and this is farnesyl residue. It has 15 carbon in it, so it has 3 isoprene units. Similarly, geranyl residue is a 20 carbon molecule, so it has 4 isoprene units in its structure. Next is fatty acylated proteins. Eukaryotic proteins can be linked to a fatty acid covalently, for example, palmitic acid. So some proteins, they are linked covalently to fatty acids through covalent bonds, and for example, to palmitic acid. Palmitic acid is a biologically common saturated 16-carbon fatty acid which is joined to a protein in thioester linkage to a specific cysteine residue. So palmitic acid is joined to the protein in thioester linkage. It means it is covalently bonded to that protein, and in some cases this palmitoylated protein is also prenylated. I mean, it has these kind of residues attached to it as well. The third one is the GPI linked proteins. So GPI, that is glycosyl phosphatidyl inositol, these groups functioned to anchor a wide variety of proteins to the exterior surface of the eukaryotic plasma membrane. So these groups are basically to attach or anchor some proteins to the outside of the surface of the eukaryotic plasma membranes. There is no obvious relationship among the numerous proteins that have GPI anchors, which include enzymes, some receptors, immune system proteins, and recognition antigens. So they don't have really obvious relationship among the different numerous proteins. The main function of these proteins is to anchor the proteins to the outside of the cell membrane. But GPI groups simply provide an alternative to the transmembrane polypeptide domains in binding proteins to the plasma membrane. Their basic function is for binding the proteins. Here is the core structure of the GPI anchors of proteins. This is the main structure, how they anchor themselves. So the core structure consists of phosphatidyl inositol, which is glycosidically linked to a linear tetrasaccharide composed of 3 mannose residues and 1 glucosamine residue. So this is phosphatidyl inositol, which is glycosidically linked to a linear tetrasaccharide. This is a tetrasaccharide, so it has 4 monosaccharides. That is why it is called tetrasaccharide. So 3 of these monosaccharides are mannose residues. These are mannose, and the fourth one is the glucosamine residue. So these are attached to the phosphatidyl inositol through a glycosidic linkage. And then we have phosphoethanolamine attached to this last mannose residue as well. And here R1 and R2, they represent the fatty acid residues whose identities vary with the protein. So they can be different fatty acid residues depending on the protein itself. And the tetrasaccharide may have a variety of attached sugar residues whose identities also vary with the protein. So it is not necessary that in all cases this tetrasaccharide has all these 3 mannose residues. It might have some other sugars attached to it as well. So this is the overall structure due to which the GPI anchors the proteins on the outside surface of the cell membrane. So that was all about the membrane, lipids and proteins, and we will study the fluid mosaic model in the next lecture. So here is a little question for you. You should find answer to this question, that what is the main function of carbohydrates in the cell membrane, why they are there. You have to find its answer, and best of luck for your studies. Here are the books which were consulted for the preparation of this lecture, and stay safe. I'll see you in the next lecture, inshallah. Until then, Allah Hafiz.